Hello, I'm Neil Spooner, and welcome to September's Patient-Centric Sampling Interest Group webinar. I hope you all had a good break over the summer while we didn't have any webinars, but we're back with a full program taking us up to the end of the year. And in fact, we've got monthly webinars booked through to the end of 2025, which is really, really exciting. But if anyone has any ideas for webinars, please just let us know. So I'm really excited and honored today to have Joleen White and Micha Levi from the Bill and Melinda Gates Medical Research Institute speaking to us today on replacing serum with dry blood microsampling for pediatric pharmacokinetics of prophylactic RSV monoclonal antibody. Before we start, I'd just like to thank all of our sponsors, without whom we wouldn't be able to make these events happen. And if anyone's interested in sponsoring the patient-centric group, again, please just connect with me. So Joleen and Micha will be speaking to you. Please remember that this event is being recorded. If you have questions, please submit them through the Q&A function. If you've been to any of our webinars before, this is a new function that's been added. So please don't use the normal chat. Use the Q&A. You'll see the new button to the top right-hand side, and we'll answer those questions at the end. So with no further ado, I'll get off the screen and hand over to Joleen, I believe.
Thank you all very much for attending. We will be talking about the global clinical development of RSMO1. It's an anti-respiratory syncytial virus monoclonal antibody that is essentially developed for prophylactic use where vaccination is not an option. So the respiratory syncytial virus has been in the news recently. It is being seen as a morbidities, especially in older adults with the when they also get COVID. But what we're talking about more is the challenges in, oh, I do not know what just happened there. Sorry, I'll come back to, all right, is that in young children, particularly infants, there are a large number of hospital admissions every year. And that over 250,000 in-hospital deaths for infants who are younger than 6 months old, with more than 99% of these deaths occurring in low and middle income countries. So this is a huge burden for those systems. And there is an urgent need for affordable, safe, and effective prevention of RSV in these regions. And so the goal of the RSMO1 development program is to develop this prophylactic monoclonal antibody in order to prevent disease in these infants, particularly in low and middle income countries. For the molecule itself, the F-glycoprotein, which stands for fusion, F for fusion, is also the site that is responsible for the binding and intake into the target cells. And of these, whoa, sorry, I do not know what is happening there, but can you see my pointer? I don't know what is going on, so I'm not even going to use the pointer and hope for the best.
I can't see a pointer, Joleen.
I took it off because when I was trying to use the pointer, the slide was jumping around on me. So I'm just going to not use it. But what you can see on this is that there is a site 0, that is part of the pre-fusion form of the F protein alone. And it is highly sensitive to neutralization. There are other antibodies that are targeting this region, and it is often a correlate of protection for the RSV vaccines, such as the Arexvy, which has been recently launched in the US. By binding this pre-fusion form, it basically prevents the membrane fusion of the virus and the target cell. In addition, the RSMO1 molecule has a YTE mutation in the constant domain that increases the half-life by decreasing the affinity for the FcRn receptor, so thereby slowing clearance. And with this extension of the half-life, there is the potential for full coverage for RSV. It is a seasonal virus. By using microsampling in our development program, there are several key features amongst the ones outlined in Spooner et al. That are particularly relevant for these infant populations in low and middle income countries. The biggest of which for us is the ethical benefits. We can obtain much smaller samples from infants if we use microsampling than if we did traditional venipuncture, allowing us to collect samples that enable effective evaluation of the drug over time, and also opens up the possibility to collect samples in a closer time frame to a clinical event, such as an RSV infection or suspected RSV infection. The patient experience is also critical, that it is less invasive for these infants than venipuncture. And by using microsampling, we also reduce, even if you're doing a heel prick in infant trials, using microtainers uses far more volume than the microsampling techniques. We also are experiencing direct cost savings in shipping from sites to our central lab. We have enough stability to show that we can ship those samples from sites to central lab ambient, which also helps because they dry while they're being shipped. And then for the indirect cost savings, we're really focused on lowering the burden of the clinical sites staffing, in that fewer staff are required for the blood collection. And we're hoping that this also leads to improved clinical trial recruitment and retention, although quite frankly, we don't have a side-by-side comparison to determine that. Another advantage for the site themselves is minimized on-site processing, and it reduces labor, resources, and potential errors than with having to process samples to serum at the clinical sites, instead of simply collecting the blood on the devices and sealing them up and shipping off to the central lab, along with the tubes for the safety labs. Of note, in these studies, we also are not processing the safety lab samples at the sites. They are simply packing up those EDTA tubes, or the serum separator tubes, and shipping them all ambient to the central labs. So that allows our samples to go in the exact same workflow for the immunogenicity, PK, and pharmacodynamics. So we did decide to go with capillary blood rather than venous. You can do microsampling, but in venous samples as well, by collecting into a microtainer, and we're doing that on some other programs. However, what we were really looking at is, for these infants, the heel prick is so standard that many of our sites are already familiar with this. And then instead, what we're doing is, instead of collecting the liquid to process and then spot, we are actually doing the whole blood directly onto the devices. And what we'll be talking about in this presentation is our phase 1 pilot in adults that was used to evaluate the ability to transition to dried blood for all of the PK in the drug program. When we talk about reducing the site sample handling burden, what we're really talking about is that until the site, we ship the samples in the regular postal service or courier, in this case, they're all relatively local. The site themselves only has to do the lancet, the collection, seal it, they're done. Whereas in our traditional serum sampling, the site is doing a venipuncture collection, centrifuging the sample, pulling the serum off, and then shipping that. We could, of course, shift the serum processing to the central lab, but we're able to take out even more steps by shipping it to the central lab. We also have nearly as much stability at -20 C than we do at -80 C for this assay. However, we are storing the samples at -80C when we get them, simply because once they're at the central lab, we actually have far more -80 stability storage than we do the -20 storage. So for this specific program, we have implemented it as the primary matrix, and in the RSMO1 101 study, we did fully matched samples, which is far overkill for an actual bridging study. We recognize that, but we wanted to take the opportunity in this adult population to really look at, in excruciating detail, the comparison between the RSMO1 PK as using blood concentrations versus serum concentrations. And by using fully matched samples, we would be able to take all of that data through the entire analytical process as independent data sets and look at the correlations across every step. A key thing to note with large molecules, particularly including monoclonal antibodies, is that we do not expect our antibodies to be partitioning into the cellular portion of the blood. So while all in whole blood, we have the denominator is all of the liquid and cellular volume. The numerator of the amount of drug present from whole blood fully goes into the serum. And so we expect that the serum concentrations will be higher than whole blood concentrations. And so we do expect to see a net bias, but across the entire set of samples, and expected to be roughly proportional to the hematocrit. We do want to calculate conversions factors so that we can incorporate serum data sets that are available from non-clinical efficacy models and other anti-RSV monoclonals into the modeling. And at this point, I'm going to pass it on to Micha, who's going to talk to you more about the study and the pharmacokinetic analysis.
Okay, so thanks, Joleen. So we performed a phase 1 first-in-human study in healthy volunteers, a healthy adult. It was a double-blind, placebo-controlled study, and the main goals were to evaluate safety, tolerability, and PK. The study had, as you can see in the slide, we had like a dose escalation phase, which consisted of 4 arms, 300 milligram arm, 1 IV, and 1 IM. And then 1,000 milligram IV and 3,000 milligram IV arm. Each cohort had like 6 active patients on RSM01 and 1 placebo. And then we started a dose expansion arm with 24 subjects and 4 placebo with a 600 milligram IM. So in this slide, you can see the correlation, and we measured PK. We had like relatively dense PK sampling, and we measured both the concentration in the capillary blood and in the serum. And here you can see the correlation between the blood and the serum. So you can see that all subjects show high correlation between the blood and the serum, and you don't see any discernible pattern when you control for sex, race, or ethnicity. And in the graphic, you can see the Deming regression that incorporates the variability into both x and y axis. So you can see that we have high correlation between the 2 matrices. In the next slide, you can see the time concentration time profile, the mean concentration time profile of RSM01 in the capillary blood and in the serum. So on the left, you can see the mean concentration in the serum. On the right, it's in the blood. And you can see the concentration time profile for each arm. I don't know if you can see the marker, but you can see the expansion arm has a less PK data, but so you can see it, and you have like more PK data in the in the dose escalation arm. So the PK is typical with half-life or prolonged half-life map. So we have a half-life, that is pretty long, around 70 days. And you see the dose linearity. When you increase the dose linearly, you can see similar increase in the exposure. The next slide is similar. So again, you see correspondence between the the serum and the blood. Here you can see the dose normalized blood and serum concentration. On the left is the serum, on the right is the blood. And when you divide the exposure by the dose, you can see that you notice the linearity, because all the graph, all the lines are collapsing on each other. You can see that the IM arm is a little bit lower. It's not overlapping the rest of the arm because of the the bioavailability. Not all of the IM goes into the system. So again, you have a good correspondence between the blood and the serum, and both of them are behaving the same. In the next slide, you can see the the individual plots, or we call them spaghetti plots. So on the left, you can see serum and compare serum and blood. On the left is the left panel is the IM. We had like two arms of IM, and on the right is the IV arm. And you can see also in the individual levels, we have like you see the same thing regardless of if it's serum or blood. You see the same kinetics. So you can see there's like every PK there, you have like few outliers. You have like some patients with a higher clearance, they cleared faster, but they seem to be consistent between the two mediums. Here we show the ratio between the PK parameters that we calculated in the blood and the plasma. So you can see here the AUC up to 90 days, 150 days, and the last concentration in the Cmax. And when you look at across all the cohorts and you look at the ratios, they're not really they're consistent. They're not really changing a lot. So there are no meaningful differences in the geometric mean ratio of the PK parameters across all the parameters in the cohorts. We also develop a population PK model to describe the data, and the POP-PK analysis demonstrated that the PK and capillary blood from VAMs and serum samples are comparable. So you can see in the table the PK parameters, and you're comparing the serum and blood. So the model that we use to describe the data is a two-compartment model with a zero-order absorption and the first-order elimination. And it was a model that fitted simultaneously the IM and the IV data. So as expected, like Joleen described the distribution, the different concentration. We expect to see higher concentration in the serum compared to the blood because the distribution to the hematocrit. So due to MAB partition to serum, clearances and volume parameters were higher in the blood. So the clearance in the blood was 0.0665 liter per day compared to serum. By approximately 40%. However, you see similar half-life because also the volume is different. So the half-life, the terminal half-life is approximately the same, around 80 days for both. And also the bioavailability is not impacted by the matrix. So the secondary PK parameter like half-life and bioavailability are not really impacted by the matrix. So we can draw the same conclusion from that. So I will hand over to Joleen now.
Sorry. In addition to the PK assay where we were measuring the drug concentration, we also did a viral neutralizing assay where what we were looking for is specifically the viral neutralizing activity that is present in the patient sample. So you could kind of think about this as a functional PK assay, but we really think of it more as an ex vivo activity assay as regarding to how much neutralizing activity is present. What you do is traditionally path is on the right, which is the serum, where you do serial dilutions with the cells and the viral stock, and then you look at the infection endpoint. With the dried blood, we do an extraction from the sample, heat and activation, and then the dilution on the left here. Then when you do the sample dilutions and calculate the ID50, you can see that you expect to see a little bit of a shift in what the inhibitory dilution is for a half maximal inhibitory dilution, based again on the expected partitioning into the liquid phase rather than the cellular phase. And what we also did, because these are adults who are presumably have a background of anti-RSV antibodies due to their natural infection, is that we did have a baseline sample that we divided the baseline sample into the individual samples at day 90 and 151. And what you can see in the next slide is that we also had great correlation for these. There's far fewer data points. However, again, we saw a good correlation between the results in blood on the x-axis and serum on the y-axis. And again, no pattern for controlling by sex, race, or ethnicity. And the Deming regression, again, was used to incorporate variability into both the serum and blood measurements. The baseline neutralization experiments also showed a good dose response, where when you look at the placebo, there's really no increase over the day 0 baseline. But then as the dose increases, you see the presence of more neutralizing antibodies as a higher ID50. And then we didn't do a lot of the earlier time points, but you do see a small or decrease in activity from day 90 to day 151. And of note, we still have fairly significant neutralizing activity, even at time points where some of the individuals are starting to go BLQ in the PK assay that Micha showed. We didn't have a single day 150 go under BLQ for the viral neutralizing assay. So in conclusion, we were pretty pleased with the success of this pilot in the adult population. We felt that we were able to successfully show that the RSM01 PK and PD activities were highly comparable with high correlation coefficients and drawing similar conclusions regardless of the data set. And that it is therefore valuable to be applying this approach to our pediatric trials, which will hopefully be starting this year in infants in Africa. We feel, and we'd like to end by thanking the RSM01 101 trial participants who agreed to be sampled both by venipuncture and by finger capillary blood collection at every single time point for every analyte. Well, the bioanalysis analytes for PK, ADA, and neutralizing activity, and several key members of the Gates MRI and collaborators. And we'd like to, of course, thank the entire Gates MRI along with the Gates Foundation for funding this trial. I'm happy to turn this over now to Neil for the Q&A, and we'll leave the slides up so we can jump back as needed for individual questions.
Thank you, Joleen. Thank you, Micha. You've left loads of times for questions here. So
Yeah,
this
we have.
Fantastic. It's a really interesting presentation. Thank you so much. We've got the first question from Gilles. Hi, Joleen. Thanks a lot for the presentation. Did you see any matrix effect, potential toxicity on your neutralizing assay using dried blood samples?
Yeah, so that was actually an initial concern of ours. And what is not shown here, but if you go to this Terstappen paper, we did do a fair bit of work trying to evaluate what the minimum dilution factor was. Oddly enough, we actually saw a cleaner matrix with the blood than the serum. I don't know if whatever might cause toxicity on the cells was just not present in the eluate, or if we were, maybe we were just extremely lucky that the cells that are used in this assay weren't sensitive to items in the whole blood. But yeah, the assay was really clean. The validation, qualification is exploratory objective here. The assay performed well in both serum and dried blood extract.
Thank you, Joleen. We have a question from Adam. I think it's more of an operational one, really. Very nice data set. Can the paper references be shared in the chat? So maybe we can do that, Adam, but this webinar has been recorded and will be available via the PCSIG website and the PCSIG YouTube channel in the next few days. So you will be able to look through the whole talk and see the references there. But obviously, if there is opportunity for Micha and Joleen to be able to share those in the chat, that would be good as well. Do you have anything further to say to that, Joleen and Micha?
No, I always put the DOIs in there because those are unique. Yannick Terstappen has published more than one paper this year, so you can go use those DOIs and make sure you're getting to the unique reference.
Yeah. Also, we have we're working on a manuscript that will describe this data in more details.
Yeah, it's in review right now, so we don't know that it's been formally accepted yet, but what we can do is on the YouTube channel is once that manuscript is published, we can put the link to that manuscript on the YouTube description as well.
So we have a question from Amit here. Other Matra VAMS device, have you tested other DBS collection devices? I don't know who wants to take that one.
I'll take that one. We did look into a couple of different devices. We took into consideration for our selection here that it is a monoclonal antibody, and protein extraction from some of the paper-based devices is a little expensive and incomplete because you do need to disrupt the cellulose layer a bit. We wanted to go with something that would be as close to 100% extraction efficiency, and the published literature to date on some of the non-clinical work had shown much closer to 100% extraction in the plastic-based microsampling devices. And so of those plastic-based devices, we were particularly interested in the Mitra device because the cost of the device was a little less expensive than the cost of the Tasso device. However, more critically was that the Tasso device at the time we started this study had not been studied in infants yet. And with using the Mitra device, we had the possibility of using lancets that were specifically designed for the infant population, including if we were doing premature infants. So the ability to take a lancet designed specifically for our patient population and then a device that was independent of the lancet was what was driving our selection for this device. On other programs, particularly our small molecule programs, we are looking at different collection devices, but I will defer that topic to maybe another webinar from the people in our group working on that.
That would be fantastic, Joleen. Thank you for a great answer there. We have a question here from Jason. I was wondering, what was the extraction method did you use? I might be missed if you already mentioned during your talk.
I didn't in detail in the talk. It is essentially a PBS buffer. For the PK assay, there's a little bit of tween in there to pull it off. But in the viral neutralization assay, it is pure PBS. Actually, you can sort of see it in this figure. It's just very tiny. It's a 1:10 dilution, so 200 microliters for a 20 microliter tip of just straight PBS. and we get 100% extraction with that. I know that sometimes you have to tweak it a little bit for different types of antibodies, but we've had great success with just a straight PBS extraction for this particular monoclonal antibody with great yield, and reproducible. It's pretty gentle when you compare it to what you have to do from Whatman paper to get the monoclonal antibodies off.
Okay, thank you, Joleen. I have another question here about, did you do any measurements of anti-drug antibodies at all?
We did. It's very low incidence, so we didn't get into it in much detail because you can't see pretty correlation plots like we have for the PK and viral neutralization. We do think that so we had a total of 5 subjects who were detected either with serum or blood. 1 patient was detected in both. 2 patients were detected in serum but not blood, and 2 patients were detected in blood but not serum. However, all 4 of the subjects were participants who had a positive in one matrix but not the other, those signals were really, really close to the limit of detection of the assays. And so we're not convinced, we don't really know from that data whether or not those patients would be considered false positives in the matrix they were detected in or false negatives in the matrix they weren't. However, what we can say is even the patient who was detected in both matrices, there was no impact on pharmacokinetics. Those outliers that you saw in the earlier slide, or actually, I'll just these outliers, they are not from ADA positive, the ADA positive subjects. So while we are going to continue to proceed with using the blood for the collection for the ADA detection, that we all know that we build in some false positives into our ADA assays. And so with these non-quantitative assays like ADA, it's a little harder to determine the correlations, but we are feeling comfortable moving forward with those results.
Thank you, Joleen. Thank you very much. Mike, I've got a question for you now. Just looking who it's from. I can't see who it's from. Gilet again. Micha, you showed for your PK assay LLOQ lower for dried blood than in serum. Wouldn't we expect higher lower limit of quantification due to more complex matrix? How were derived the LLQ? Thanks.
So the LLOQ came from Joleen's group, but it's we have like a lower concentration in the blood compared to the serum, because so this is
yeah. And like I mentioned for the viral neutralizing assay, it's actually ended up a cleaner matrix somehow. I honestly have no good explanation for why an extract of whole blood is cleaner for us to work with than a extract than serum, but it just was. I'm assuming that was just pure luck and that it was related to our critical reagents that we were using, but the numbers, you saw them. It's a lower LLOQ in the dried blood assay than serum.
Thank you, Micha and Joleen. I have another question here about basically, obviously in very young infants, you get some different hematocrits, and they can change quite a lot in the first few days and weeks of life. Does that impact your quantitation? Did you take that into account at all?
So this data is from adults, so it's not impacting this data. But we have a subsequent study, a PK study, that we're going to start very soon in infants, actually newborns, and we're gonna measure PK in both matrices to compare the impact of the potential changes in the hematocrit with age, and we expect there will be some impact. Honestly, there's no good data on how hematocrit changes in infants, especially in Africa, with age. So we will get very useful data from this subsequent PK study.
Yeah.
But we're monitoring that. Yeah.
So it sounds like some very useful data on a number of fronts there, Micha. Look forward to hearing about that in the future.
Yeah, so of course, we don't know how to extrapolate the PK from adults to infants, with these long half-life antibodies, which is important. But also the other impact is comparing blood and serum in this population. Yeah.
Yeah.
Obviously, we don't have quite as many time points in serum in the next matrix, but we did make sure that we do have some at the earliest time points where the infants are younger, and current literature suggests their hematocrites will be highest.
Okay. Thank you. Another question here, and it's a question that comes up a lot when we're talking about these alternative approaches for collecting samples, is simply how well is the data accepted by regulators in different parts of the world?
So we know that the FDA and Europe and Japan as the lead countries in ICH have included in the M10 guidance their expectations of assay validation, and we have met those. With regards to the PK data sets, however, we have been focusing primarily on the regulatory authorities in the countries where we're doing the studies. So when we had submitted a briefing book to SAPRA, which is the South African, can't remember the whole acronym, but basically it's the South African Health Authority. We explicitly told them that we were planning on running everything in the microsampling, and one of their key comments was to make sure that we were, which we replied with the microsampling as well, was make sure you're being really conscious of your total volume you're collecting in infants. We also have found a reasonably good number of publications, which we presume were about studies submitted to regulatory authorities about doing the African studies with microsampling and accepting it. Well, I don't have personal experience with a submission that has used blood as the only matrix. I know we are not the first to do it, but I haven't come across anybody telling me that those data sets weren't accepted. I think the biggest challenge is really on Micha's side, where when we're building models using our final data, it will all be in blood, but on the way there, where we're incorporating data from other similar molecules, all of that data is going to be in serum. So we have been thinking about how do we do that in conversion. And you can see that a little bit on this slide, where we have an estimated efficacy threshold for serum of 6.8, but you don't see a line on the blood side because we don't have a blood estimate of efficacy at this point. So you can kind of see that emerging as we go through our system, but we will eventually have enough data to drive this forward.
Just a follow-on question to that. Was your non-clinical data in serum, or was that in whole blood?
Our non-clinical data is all in serum. That was just as a anecdotal why that switch didn't happen for non-clinical is the non-clinical was done prior to the COVID pandemic. And when we hit a delay in the program due to the COVID pandemic, because obviously to us, maybe not to you, is everybody who was studying respiratory viruses for a couple of years was working only on COVID and stopped doing their work on RSV. So we had a multi-year delay in our clinical program, and we took that time to really go through and invest in being ready to do this with dried blood sampling. Instead, get internal buy-in, have the discussions about what it would take to incorporate that into a phase 1 trial in adults. And so it was the adults study, first in human study, that was our first opportunity to actually incorporate the microsampling. We've done future study other molecules where we collected both and in non-clinical, but that did not, for some reason that assay didn't work as well in in humans. So we are relying more on the serum data in that molecule, which also was not being done in infants.
But I think following up on Neil's questions, I think it's valuable to start, as early as possible. If you decide you want to do like blood PK, in humans, so you should have something comparable in the preclinical models, because it's becoming like a complicated to compare, different matrices in different species. So
We
it's
did do a pilot with one of our pharmacology models in a different disease indication, where we had the academic lab collect on the Micha devices. It was another antibody, and they found them very easy to use because their pharmacology endpoint was also blood sampling. So at the same time that they were collecting a small volume, they would just add another little sample for the PK, and we got some really beautiful profiles out of it. But that work is going to be presented elsewhere. So we're just speaking to it. Is we have determined it's feasible, but we are still working on figuring out which programs are going to use which devices, as well as if we do the transition prior, if we can do it as early as discovery, that's really where the biggest benefit to the PK modeling is, because that's where we have our efficacy models. The tox studies don't really have the efficacy endpoint. So while the PK data is used in the early modeling, as soon as you start getting human PK, it drops out. It's the efficacy modeling that I think is more critical to try and get the matched data on. And that's, now you're talking about assays that aren't even necessarily fully validated for the drug concentration. So
Yeah.
It's tricky.
Yeah, understood. No, that's really interesting. Just reminder to everyone listening, if you do want to submit a question, please submit them through the Q&A function. I have one more question available here. It's my own question, actually. Did you get any, did you collect any data through surveys as to what the clinicians and the patients actually thought of this alternative approach to collecting blood samples?
No, we did not. We know that that is something that is of great interest to the field. We are getting sort of anecdotal information as we talk to clinical sites. For the infants, it's a little bit different. People are very used to doing heel pricks in infants instead of venipuncture for the youngest infants. For other pediatric indications, they're already comfortable doing the butterfly needle venipuncture. And so we've really been deferring a lot to our sites as to how it's collected, but everybody appreciates the lower volume, whether we're sampling directly from a lancet on the skin or sampling from a microtainer that has been collected from venipuncture. The lower volume requirements has been of great interest to all of our sites. It's just whether or not they're ready to go to the capillary blood versus the venous blood is still a little bit more in flux when we talk to our clinical sites. But so far, the collection is pretty well received in the infant, because they've been doing heel punctures all along with capillary blood. So switching to a lower volume is actually a very conservative move for them. And for our first study that's in South Africa, the Micha device is already registered with their medical device. So that's not an issue. We are still trying to figure out the logistics of running it and running studies with that device in countries that do not have all of these devices already registered. But so far, those are also the sites that prefer doing the venipuncture with microtainer, followed by spotting from the microtainer itself.
It's complicated.
It is, but we're learning as we go. There's a lot of logistics challenges with running trials in low and middle income countries that aren't necessarily present in the high income countries. but we think it's really worth it to make sure we're generating data in the populations that we are aiming to treat with these therapies.
It's fantastic, and thank you so much for sharing those kinds of experiences, because it's those details that's going to help other groups that are wanting to implement these technologies in whatever populations. There's always going to be complexities, and sharing our experiences is through webinars such as this and through publications is going to help everyone. Just on the questionnaire side of things, just for yourselves and for the audience out there, the patient-centric sampling interest group has developed a patient questionnaire that is currently being reviewed by our patient advisory group, and we're looking to publish that in the next few weeks on our website. And the idea is that's a modular questionnaire that people can hopefully all use the same or similar questions, which will not only make it easier to add them into your protocols, but also if we're all asking related questions, it means we can anonymize and pool our answers and give a much more powerful data set. So watch out for that in the next few weeks, hopefully.
I have a question for you, Neil, then. Does it have any questions for caretakers, for younger patients who can't actually answer the questions themselves?
It does, from my memory. I've not seen it for a little while, but it does, Joleen. But once it goes out there, if there are things like that missing that you think should be in there, please let us know, because it's designed to be a live document, that if we are missing something important like that, and I think it is in there, I just don't remember, please let us know, and we can look to add that in again. So I'm not seeing any more questions coming in. So just last chance for questions. If not, could I just share my screen for a moment to advertise the upcoming webinars?
Sure, go for it.
So just advertising a little bit of the upcoming webinars. So coming up on October the 2nd, we have a webinar with multiple presenters based upon a workshop we had in June, attended by about 50 people, focusing on incorporating patient-centric sampling into multi-center clinical trials. So very much related to what Micha and Joleen have been talking about today. We'll be talking about some of the challenges of doing this and what the community thinks are how to overcome some of those challenges. And there's an opportunity in that webinar to vote on what activities we should be concentrating on to help overcome the barriers to adoption of these technologies for multi-center clinical trials. And then on October the 22nd, so we're breaking our rule, we're doing 2 webinars in October. We have opportunities and challenges in mobile phlebotomy and self-sampling for precision chemotherapy dosing in clinical trials with Ellen and Sophie from Karolinska. So some exciting opportunities, and keep your eyes open for advertising for the rest of our webinars. As I say, we're now booked through to the end of 2025, but if you have suggestions for further webinars, please let us know. Just quickly, the patient-centric sampling interest group, if you're not already a member, want to learn more, please use those QR codes to take a look at our website. And the QR code on the right will take you to our LinkedIn group, where there is about 450 of us sharing information about patient-centric sampling for a variety of different applications. And finally, I'd like to just thank our partners and our sponsors again who enable us to do this. And again, I'd like to thank Micha and Joleen for a fantastic webinar today. And I'd like to thank you for all attending today. Very interesting, and welcome back after the summer. Thank you all.